This week, we are going to learn about fungi, one of my favorites. Such a weird and interesting group with lots of fun and exciting types of ecology. This week, we're going to focus on understanding the importance of fungi to plants, to animals, to us, and also to understand how they make their living and their unique morphology, which is really fascinating and very different than a lot of the organisms that you're used to, and understanding fungi reproduction. It's important to remember that there are a great deal of different things that fungi are associated with. They have nematode trapping fungi. They have fungi that are introduced by insects like beetles. Fungi are associated with some pretty dreadful diseases of things like our bats and our frogs and amphibians. They're also associated with lots of different types of interactions with plants. And then over here you can see that there are fungi that are grown by ants as a food source. So lots of interesting things happening with fungi. Remember that they're a very diverse group. They're saprophagic, which means that they're going to live on decaying organic matter, and that there's lots of situations where they're going to either be saprophatic or decomposers, or they're going to be parasites or mutualists. Don't forget that they are also more closely related to animals than they are to plants, which is often what people forget about because they're slow moving, if not no moving, right? Compared to animals. They seem a lot more plant-like to us, but actually they share a lot of qualities that are associated with animals. There's a lot of evidence for these close relationships, including their DNA sequence, a lot of associations with things like chitin, which are associated with insects as well. Their animal flagella, their flagella are similar to animal flagella in the chytrid group, and they also store glucose as glycogen, just like our animals do. Remember or get yourself acquainted with the phylogeny. I'm not going to ask you to draw these phylogeny out, but I do want you to understand the relationships with them and whether they're monophyletic or paraphyletic. You can see that when you get up to the chytrids and the Zygomycetes, they're a big mess. Basidiomycetes, Glomeromycota, and our Ascomycota are going to be a little bit more, a little neater of a phylogeny. And here's just a close-up of that, and you can see we do have some monophyletic groups here based on primarily their reproductive structures. I also want to remind you of making sure to get acquainted with their importance in terms of the nitrogen cycle, so make sure to read through that. And make sure to understand the structure of fungi. They come in very unusual forms, either the single cell form that many of you might be familiar with because you're baking lots of bread. So this is our yeast. And then we have our multicellular fungi that are associated with all that underground mycelial network. In addition, we're going to talk a little bit about their unique life cycle associated with those hyphae. So we have some different forms that are going to be new to you that are filamentous. They have an interesting anatomy that we'll talk about in more detail and discuss, and you'll watch videos about it here. It shows you a little bit about their hyphae and this septum associated type of characteristic, whether or not they have that septum or they don't have it. And then we have some close-ups of what that looks like when you actually look at them under the microscope. And here's some more pictures associated with what this type of structure looks like. So this shows you the dense hyphae, spaced hyphae associated with the reproductive structure. And this shows you whether or not you see it's coincidic or it's has a septus. Remember, coincidic is aseptate. And there are a lot of different interesting reproductive structures, which really gives them all their name. And here are some examples of what those reproductive structures look like. The sporangium, or our bread mold that you probably all grew on your bread. I won't expect you to know all the life cycles in great detail, but I do want you to know the generalized life cycle by heart. I also want you to familiarize yourself with the different major groups. So our chytrids, our Zygomycota, so our bread mold, and our Pilobolus, which is a really cool different group attracted to light or moves towards light. Here's an example of our Basidiomycota, the Boletus, and our Ascomycota, our cup fungus, which are very interesting species. So I want you to familiarize yourself with the different groups and ask yourself about what their spores look like, sexual, asexual, do they have specialized structures, and where their genes from associated with these specialized structures. Ask yourself about alternation of generation this week and how we see it in the fungi, which is different than how we see it in the plants. And one of the most interesting things about them is their relationship with other organisms. So ectomycorrhizal and arbusculomycorrhizal or endomycorrhizal, you might hear it. And so these are relationships with plants that we're going to talk about and look into. Very, very interesting. Probably the main innovator for why plants moved to land had to do with their relationship with fungi. Reading and understanding the relationship of lichens to fungi, or how lichen are fungi. They're composite organisms or fungi and some sort of photosynthetic organism, more than one usually. And then you're going to once again just think about these different groups from our chytrids, which are generally pathogenic, our Zygomycota, the ones that are associated with bread mold, and then here are our cup fungi again, our Ascomycota or sac fungi, and then Basidiomycota, the ones that you're very familiar with, which are the classic mushroom. And then last but not least, you're going to have your Glomeromycota, which is an interesting one called club fungi, mostly very tiny types of structures. So this is what you're going to cover this week. Don't forget about your research projects this week. And please do come by my office hours if you have any questions or would like to discuss your research project. Looking forward to seeing you this week. Have a good one.